Good morning. Today, I'd like just to review a couple of recent publications that have come out in October 2025. Part of my alerts video. This is number 3. The first one came out from Daily Mail, but asking whether the gender ratio is the same with IVF and to the natural population. And in this study, they believe that 50% of all babies are males, and now doctors know the surprising reason why. So this is published in the Daily Mail, and you can see the URL down here. So it goes something like this. Most expectant parents would believe their chance of having a baby or a girl was 50-50, but those having IVF are more likely to welcome a son, according to scientists. Experts have revealed that male embryos grow slightly faster and are therefore more likely to be selected for transfer to the womb. And it could explain why the odds of having a boy through IVF are as high as 56%, or 56 in every 100. Now, some fertility clinics also offer the use of AI as part of the grading process, using time-lapse videos made as the embryos develop. And Dr. Neal's team carried out a study to determine how likely male or female embryos were to be chosen, both by the doctors or by 2 different AI systems. They tested 1,300 embryos where the gender or the sex of the embryo was already known from genetic testing. Analysis revealed that when the doctors or the scientists did the selection, they rated 69% of males as a good quality compared to 57%, mainly because of the rate of cell division. So one of the AI systems had a slight sex bias towards boys, while the other AI system rated males and female embryos the same. So what that's basically saying is that if you use the rate at which embryos develop to blastocysts as a guide, since male embryos develop slightly quicker than females in some cases, that there's a natural bias of an embryologist to pick out the fastest growing embryo, since that's where all the pregnancies come from. But some AI systems have been tweaked to take that into account and to randomly pick the best embryo regardless of gender. Okay, the second one is, which I thought was an interesting one. It appeared in Gynecology and Endocrinology. It's in December. That's even before this, the talk. This one is called Implantation of Single Euploid Embryo Transfers, the Role of Blastocyst Expansion Grade and Early Serum Bed HCGs. And this, I think, is fairly interesting. Basically, they concluded that completely hatched blastocysts, CHBs, with expansion grade 6 were associated with lower early serum beta HCG levels compared to partially hatched blastocysts with the expansion of grades 4 and 5. Incorporating the expansion grades into prediction models could enhance decision supporting tools for IVF. What's interesting here is that there is a drift, particularly with using non-invasive genetic testing, to allow the embryos to grow out that extra day, to allow more material to be incorporated in the spent culture media for genetic testing. So there is a push for some clinics not to put the best, fastest growing embryo back on day 5, but to carry them all over to day 6 to get more genetic material to avoid having to do a biopsy. But what they're saying here is that if those embryos hatch completely, then their pregnancy rate is poorer than if they were partially hatched. So the debate is going to be in the future whether you should always put the embryo back on day 5 that's growing the fastest, and particularly if they're starting to hatch, that is where the best pregnancy rates are. But if an embryo is already hatched, then basically their pregnancy rate, as marked by the beta HCG, is kind of similar to an embryo that has not yet hatched. Watch this space. Another interesting one, this is continuing, there's quite a few articles now on potential overestimate of abnormalities in PGT-A. This one came out in Nature. Live imaging of late-stage pre-implantation embryos revealed de novo mitotic errors, and also a summary of it in BioNews. It goes something like this. PGT-A may overestimate impact of embryo abnormalities. Don't worry about me. A new imaging technique allowed scientists at the University of Cambridge to observe that some genetic errors appear late in embryo development than previously thought and were present only in cells destined to become the placenta. This means that some embryos flagged as abnormal may still have a viable inner cell mass, and that could influence how clinics select embryos in the future. They discovered that some chromosomal abnormalities arise after the cells that would have gone on to become placenta have already diverged from cells that eventually become the body, that's the inner cell mass. Biopsying cells taken to be representative of all the cells in the embryo, therefore you have an inbuilt error. However, new research shows that embryos can have changes in these cells that are not present in the parts of the embryo that goes on to the fetus, the inner cell mass, therefore giving a potentially misleading picture of the embryo's overall viability. As a result, the study suggests some embryos may be discarded unnecessarily, potentially affecting patient outcomes and costs, etc. Okay, so the upshot of this is that, as we know, PGT-A only samples cells in the trophectoderm and leaves the inner cell mass cells separate because, one, you don't really want to be biopsying that. However, cells in the placenta seem to develop a higher rate of abnormalities, and therefore when you test it and you're showing that it's heavily mosaic or it won't be aneuploidy, you're looking at the degree of mosaicism. But generally speaking, embryos with less than 50% mosaicism are transferred with good outcomes, and embryos with more than 50% mosaicism are usually not. However, it's suspected that some of these may still have a euploid inner cell mass and therefore give rise to a baby, which is normal. In other words, some embryos have been discarded based upon biopsying the trophectoderm, which has an inherent flaw in it. And lastly, donor eggs drive most IVF success for women over 43. Shifting reproductive windows, the contribution of ART and egg donation to fertility rates in the UK. The one in Medical Express and in obstetrics and gynecology. Okay, it's all about the women's age. Among those aged 43 and above, success rates remain below 5% when using their own eggs. In contrast, more than 1/3 of the treatments using donor eggs, which had come from younger donors, are now successful across all age groups. In 2018, these accounted for more than half of ART births aged 43 to 44, and more than 90% of women aged 45 to 50. So the authors were saying our findings indicate that fertility recuperation at advanced stages is highly unlikely to succeed with IVF using a patient's own eggs. Egg donation and egg freezing can improve the chance of conception, but they also have important limitations, making them insufficient to fully offset the fertility loss associated with delayed childbearing. They also emphasise the importance of broader policies that will make it easier for those who wish to have children early in life to do so through greater economic security, workplace flexibility, and family-friendly support. Such measures would help reduce the risk of age-related infertility and the growing reliance on complex reproductive technologies later in life. Well, once again, this is a debate which is going on whereby women in their middle to late 40s, 42 to 48, are still wishing to have children, but by that time, there's very few eggs which are really healthy. Therefore, their chance of pregnancy is very low. But their potential for pregnancy remains the same if they use eggs which have been recovered from younger women. And so what's interesting now is that clinics are starting to treat women in their mid-40s using donor eggs and having good success. But it also means that women in their mid-40s are starting to become pregnant, and those babies will be reaching maturity, like finishing school, etc., when the women are in their mid to late 60s. It will also start to distort the figures because the risk here is that clinics will start to report pregnancy rates in mid-40s of 30-40%, and women of that age will start to think that that applies to them and not realising it's to using donor eggs. And so the thrust of this point is to say this works really well for older women, therefore we should make it easier for older women to access younger eggs. But then they go through and say, well, what we should be trying to do is get women to have babies earlier. It's all very confusing. And lastly, it's a man's turn. Older men's sperm carry more mutation and they may turn harmful. Sperm sequencing reveals extensive positive selection in the male germline. Okay, older men's sperm carry more mutation and they may turn harmful. Now, this is interesting because in the context of the previous argument where older women are turning to IVF to have late 30s and early 40s and mid 40s, they also, by and large, have husbands who are of similar age. And if they're trying to have a child using the husband's own sperm, which they will have, even if they're using donor eggs, that the age effect on women of having few good quality eggs also has an impact on their older husbands, where they may carry more mutations. So even though you can get fertilization and pregnancy, there is an increased genetic risk that those babies may carry harmful mutations. So it goes something like this. Potentially harmful DNA mutations can amass in men's sperm as they age. New research has found, which may in turn impact the number of mutations passed on to children and the risk of disease in the next generation. Mutations occur in DNA where cells replicate and arise either by random chance or by environmental stress. Not only do mutations occur at higher rates in older men, the data shows that some are selfish, giving the cells that carry them a growth advantage so that they replicate faster or outlast other cells in the testes and gradually take over. Many of these mutations have previously been linked to developments of disorder and cancer. And it's a lovely graph to show. They analysed 81 sperm samples from 57 healthy men, some of whom were twins, a factor which helps separate the effects of age on sperm DNA mutations from inherited genes. And what's interesting is around 2% of sperm from men in their 30s were found to be carrying disease-causing mutations, which jumped to 3 to 5% for middle-aged men and older men over the age of 43. And boy, that makes me feel old. By 70, that's me, and by 70, an average of 4.5% sperm have potentially harmful mutations. Okay, so that's enough for the new publications. I hope they found them interesting. Just a welcome disclaimer. My name's James Stanger. I'm an embryologist with more than 50 years' experience. I'm not a medical person. This information is not medical advice, but it may help you in knowing what questions to ask about your treatment with your doctor and maybe to help in understanding the advice you receive. If you like what I'm doing, click subscribe, or you may like to leave a comment and click like, or you may like to make a donation to my GoFundMe page or subscribe to my Patreon CPD program, which are still undergoing development. And I need to acknowledge that these videos are paid for by a sponsor, which is my own website, FirdAid, which is a primary sponsor of the channel. It's an online EQA program, a training program in IVF for andrology and embryology. And that it's interesting because if you're a client, you may benefit from knowing that the skills of your laboratory staff are under constant review and assessment if they subscribe to my FirdAid channel. I run EQA schemes in andrology and embryology. You can enroll as a trainee or a professional, small clinic or large clinics for training or competency or accreditation. And that, my lot, is your lot for the week. Thank you. IVF, I know, is a personal journey that involves the action of many staff and techniques and technologies, and it helps to understand all aspects of that journey. I hope this information may be of value. If you like the information provided, please tell other clients about this service and please subscribe. I'll say goodbye and see you in a couple of days.